Hello, everybody. It's Gina again. This is video number 3, and in this video, you're going to have the enjoyable experience of learning how to do a monohybrid cross, a regular old Punnett square. Maybe you remember it from high school, and maybe you don't because high school was 20 years ago for some of you. So we're going to start at the beginning. And we're going to do monohybrid crosses. We're not going to do any dihybrid or trihybrid crosses. That would include crosses that, like a dihybrid cross would cross 2 traits, like, for example, flower color and pod shape, right? That would include, you'd have to have 16 boxes to do that. We're just going to do a monohybrid cross, 1 trait, like flower color, can you roll your tongue, that kind of thing. It only has 4 boxes, so it's pretty simple. One of the things that I have you skip, if you're using the textbook, which you don't have to, but if you need to go back and read stuff, if the handout and the PowerPoint's not enough for you, if you're using the textbook, which, it's a good idea, there's a whole section on probability. Skip it. We don't need to do any of that stuff. We don't need to add the math. So just something that you need to make sure you understand is that if you have 4 possibilities, right? Your Punnett square will have 4 boxes. If one of the offspring out of those 4 possible offspring that are made has the genetic trait you're looking for, that would be 25%. If 2 out of 4 have the genetic trait you're looking for, 50%. If 3 out of 4 have the genetic trait, that'd be 75%. And if 4 out of 4, or all of them, have the genetic trait we're talking about, that'd be 100%. That is the extent of the probability and the statistics that we need to do for this chapter. So don't worry about the whole section on all that math and stuff. We're not covering that. We don't need to do that here. Okay, so I'm going to share my screen and I'm going to show you how to do a Punnett square. So hold on. There you go. Okay. So here is, we're just moving on along through the PowerPoint, and we're going to do a Punnett square. I am actually going to do a Punnett square right on this page right now, and I'm going to show you Gregor Mendel's original cross. I'm going to show it to you right on this screen here. I'm going to do it right at the bottom, and I'm going to give you information on how to sort of the basics. Let me see if I can. I don't know if I can create a whiteboard. Let me just try to create a whiteboard. So okay, so we have our whiteboard here, and I'm just going to show you how I'm just going to show you with the Punnett square Gregor Mendel's original cross. Okay, so let's get this rocking here, and I think we're good with all the colors that we have. So I'm going to just, when we do a Punnett square, we show it with a square. Let me just turn this off. Okay, we show it with a square. And the square has 4 boxes. Now, just a couple of things. A Punnett square is going to represent, it's going to simulate sexual reproduction, not asexual, sexual reproduction. So that means the bringing together of a sperm and an egg, right? So while this doesn't matter which direction you do this, just for this example, I'm going to say these are gametes, right? These are the eggs, and I'm going to say this side is the sperm. It doesn't matter. You can flip-flop them. It doesn't matter. But I'm just going to say that. So we're going to talk about what happens when the egg hits the sperm. Okay, what happens when the egg hits the sperm? And that's what I mean by it represents sexual reproduction. The other thing we want to remember is that egg and sperm are haploid. Okay, let's not forget that they only have 1 copy, 1 allele from the in each of the egg or the sperm. There's only 1 allele. So if the individual is like, let's say big B, or big B, let's say big B, little b. If the individual that's diploid is big B, little b, the eggs that that individual makes would either be, the egg could either have a big B in it or a little b in it, but they're not going to have both because eggs and sperm are haploid. They only have one copy of the chromosome. Okay, so eggs and sperm are haploid. But so those eggs and sperm go on the outside of the boxes. Okay, the inside boxes represent the offspring. Those offspring are diploid, right? So let's not forget, they're diploid. So in each box, there should only be 2 letters, big B, little b, right? Okay, so they should be diploid, 2 letters. Okay. I think that's good. Let's give it a try. So if we remember, if you go back a couple of slides, you remember that Gregor Mendel crossed 2 purebred or 2 true breeding pea plants, that one was purple and one was white. Okay, so we know that just because I'm giving you the information, we know that purple is dominant, right? And since they were true breeding or purebred, they were homozygous for both of them. Okay, so we know that in our original example of Gregor Mendel's purple flower crossed with white flower, we know that the parents, one parent was big P, big P. We're using P because purple is the dominant trait, so we're using P. And we know that the other parent was little p, little p. I'm using script P's or cursive P's, makes it a little easier. So this is the cross we're doing, big P, cross with little p, little p. So I'm just arbitrarily picking one of them to be the eggs and one of them to be the sperm, just arbitrarily, it doesn't really matter. So let's say that purple was the egg, so big P, right? And we're separating them out at the top there. One goes on one side, one goes here, right? Because they're haploid. If we put them together, I'm going to do it just to be wrong. This is wrong, what I'm about to do. If you put them like this, that makes the eggs diploid. That is not good. That is not what we wanted to do. So we're going to get rid of that P. That was kind of nice. All right, and then we'll make the sperm the white flower. So little p, right? They're separated. They're not together. We did not put 2 little p's in this area here. Okay, we didn't put a little p. We separated them out. Okay, so we've set up our Punnett square for our monohybrid cross of Gregor Mendel's original pea plants, purple versus white. And now we're going to simulate sexual reproduction. So imagine, I'm just going to draw, like, let's imagine this is an egg. These are eggs, right? And let's imagine that these little suckers here are sperm. They're little flagella, right? The only flagellated cells in mammals or animals. All right, so that's a pretty bad drawing, but that's okay. All right, let's make believe this egg, let's change the color on this. Let's change it up here. Let's make believe that this egg is fertilized by this sperm. Okay, if that egg is fertilized by that sperm, the offspring here are going to be big P, little p, right? Because this egg is big P, this sperm is little p. They're going to come together and restore that diploid number because the pea plants are diploid. Okay, let's keep going. Let's do the next one. Let's make believe that this egg here is fertilized by this sperm. And that would make a big P, little p offspring. Let's keep going. Let's fertilize this egg in this box with this sperm here. Okay, that's going to leave us, if you're following along, that's big P, little p, big P, little p. Okay, let's keep going. Let's do this last box. All right, so this egg drops down and this sperm comes over to fertilize it. And that gives us big P, little p. Okay, that's a mess, but it's good. It's a good mess. Okay, and the way I always ask this question in your handout, you'll notice is you're always going to see a little box that's going to say, or a little section, maybe not a box, but a little section that says genotype percent phenotype. You're going to see that written like that a lot with a line underneath it. When we write down, we have to kind of evaluate the cross, like what are the outcomes, the probability of this cross. Okay, I'm going to do something that might upset you, but it's okay. I'm going to do it anyway. Okay, I'm going to draw this again. I'm just going to clean it up. Okay, so I'm just going to clean this up so it's very clear to you what's going on here. Big P, little p, little p, and we cross these. Here we go. I just wanted to clean that up. Okay, so now we have genotype percent phenotype. Oops. Now, genotype is the allelic combination. Phenotype is a physical characteristic. You know that purple is dominant over white. Okay, that is something I have told you. I have to give you that information. Okay, so genotype, when I look inside the boxes, well, I only see one genotype. Right, write it down. Hit pause if you're trying to figure it out, because I'm going to write it down in a second. But there's only one genotype. The only genotype that I see is big P, little p. Okay. So if all of them, all 4 out of 4 are big P, little p, that's 100%. And now the question is, well, what color are the what color will the plants be, the flowers be, if they are big P, little p? Well, since the dominant allele is present, the dominant trait is going to be expressed, so they're all going to be purple. Okay, that is the answer to this question. The question being, what is the phenotypic and genotypic outcome of the cross of a purple flowered plant and a white flowered plant? Okay, let's do another. I'm going to do another problem that's not in your handout. I'm just going to do it anyway. I'm going to say now, well, let's keep doing what Gregor Mendel did. Let's cross this F. So this is the F1 generation, right? This is the P generation. This is the F1 generation. Let's cross the F1 generation. Let's take 2 of these plants, 2 of these big P, little p plants. Let's cross them. Let's see what happens if we cross them. Okay. So from the parent generation, we got 100% purple, just like Gregor Mendel did. We're just showing you with a Punnett square. Gregor Mendel couldn't do a Punnett square. He didn't understand DNA, right? But you understand it. So let's cross the F1 generation. So I'm going to clear this out. And so we're going to cross the F1 generation. So the F1 generation was big P, little p, crossed with big P, little p. Let's do that problem, okay, and see what happens. Once again, purple is dominant. We're going to draw a box. Give ourselves 4 boxes. Okay, this one's it's actually a little, well, it's not easier, and it's the same. So I'm going to say one of these is going to be the eggs. One of them is going to be sperm. Doesn't matter because they're both the same. So big P, little p, big P, little p. Okay, I would hit pause if I were you and try to do this out and get the genotype, phenotype out before. I would just hit pause, see if you could do it on your own. And then start it again and watch me do it. So hit pause if you have some confidence in what you're doing right now. If you don't have as much confidence, just play it and watch it because you're going to have a lot of opportunity to do Punnett squares. Okay, so just hit pause if you're ready. If you're not, here we go. Okay, so we're going to put all the alleles together. So in this box here, this first box here, we're going to get a big P, right? This P and this P. The second box here, we're going to get big P, little p. Always put the dominant allele first. Just once. Okay. This box here, big P, and this box here, little p, Okay. If you don't know how I put those together, go back a little bit, like a minute or 2 in this video. Now, genotype, phenotype. So you're going to see genotype percent phenotype. All right, so now we got a smattering of genotypes here, so that will equal a smattering of phenotypes. So I'm going to start in my top box here, in this very first box. So in that box, I have big P, big P. Okay, that's one of my genotypes. I don't have that in any other box. So in these two boxes here, I have big P, little p. And in this last box, I have little p, little p. All right, so I have 3 different genotypes. Variation. Okay. When I look at if I remember my Mendelian genetics says that if the dominant allele is present, the dominant trait will be expressed. So since these two here, big P and big P little p have the dominant allele, that big P, then I know that they are going to be purple. So big P is 25%. Big P little p is 50%, right? Because 2 of them, 2 out of 4 is 50%. And little p is 25%. Now, big P and big P little p, like I just said, they both have the dominant allele, so they'll both be purple. Okay, so 75% of the flowers are purple. And then little p, since the dominant allele is not present, the gene, the bit of DNA that says how to make purple is not there, these plants are going to be white. So this will be white flowers. Okay, so that is Gregor Mendel's F2 generation. And he did it over and over again, and he always got this ratio of 3 to 1. 3 purples to 1 white, or 3 dominantly expressed traits to 1 recessive expressed trait. So that's what Gregor Mendel was able to give to us in this idea of Mendelian genetics. When the dominant allele is present, the dominant trait. Okay, so let's go back to the handout. And we're going to share. I'm just going to hit stop sharing, and then we'll do it again. All right, we'll go back to this. Okay, here we go. And we're going to just show you, this is exactly what we just showed with Gregor Mendel. That was the cross we just did. We just crossed the F2 generation, and it's exactly the same as what we got, 3 purples and 1 white. Same idea. All right, so let's do another practice problem. And once again, when you do a Mendelian problem, one side's going to be the sperm and one side's going to be the egg. And since sperm and egg are haploid, there's only 1 letter at the top of each box or on the side of each box. You can see that in this image here, just 1 letter. Right? And then you put them all together. So that this isn't the practice one in your handout, but this is just a practice one to show you how this works. Okay, here's a video of somebody teaching you how to do Punnett squares if this video doesn't work. So there's a video in the video. All right, so here you go. Just hit pause and try to do it. Right, try to do this problem. So this is an example where you have to use your vocabulary skills. A homozygous dominant dimpled individual with a homozygous recessive non-dimpled individual. So hit pause. Try it. See if you can do it. Okay, so you must have done it already, and now you're back. So in the next slide, so basically I'm just setting this up for you. So if I see these vocabulary words, homozygous. Right? Homozygous means same. But this is dominant, so it must be the big letters. So we're going to use D. So this would be big D. And then with a homozygous recessive individual. So homozygous, same. But this is recessive, so that's the little letters. So little d. All right, hopefully you set it up that way. If you didn't, go back and fix it. So you should be crossing big D times big D, little d. Right? Okay, so this is how it gets set up. Big D, cross with, and that's arbitrary. You could switch egg and sperm, it doesn't really matter. Little d, and you put them together. You could do it one way or the other. You put all the D's in the top, the sperm in first. You put all the eggs in after, however you want to do it. Stimulated sexual reproduction, and you can see that the offspring from this cross, the offspring from this cross are all big D, little d. Right? So you would, under genotype, you would put big D, little d. All of them are big D, little d, so it's 100%. And the phenotype is dimples, because in every single one of these offspring that you make, only one offspring is actually ever produced. This is the probability, right? So if you had these two parents, egg donor, sperm donor, if you had these two individuals, they would only produce dimpled individuals, right? Because the dominant allele is present in every possible possibility. Okay, so that's how you do. If I the whole one easy, this is an easy. That was an easy one. Right? So there are two more examples in your handout. So hit pause and try to do both of these, example 2 and example 3, and then I'll show you how to do them. So hit pause and then come back to this. Okay, so I imagine you're back, and we can see here that when we read the question, what are the possible offspring, right? So these 4 boxes are offspring of the mating of a homozygous dominant tongue rolling individual with a heterozygous tongue rolling individual. Okay, so we have to decode homozygous dominant. Homozygous means same, and we're going to use T. So if it's dominant, big T, and we're going to cross that with an individual. Oh, we got heterozygous. We haven't had a heterozygous one. Heterozygous tongue rolling individual. So that's different. Hetero means different. So that means we're crossing it with a person who's big T, little t. So if you didn't do that, go back, hit pause, go back and fix it. And now I'm going to show you the solution to one. So you're crossing big T with big T, little t. Okay. If you switched them, if you put the big T on top, same, no big deal. It's exactly the same problem. Doesn't matter where you put them, just as long as you separated them out, right? That there's a big T here and a little t here. If you put big T here, let's say, or big T, little t here in one box, then you did that wrong. Okay, then that's not good. But you got to fix that. All right, so cross them. We'll put them. We're going to cross the gametes. Right. So we got. Put them all into the offspring boxes. Oh, look at this outcome. This is a new outcome. We haven't seen this one yet. Right. So if you followed along and you wrote your genotypes and phenotypes down, you would see that it's like a 50-50 option here of genotypes. Big T. We see that here. These are all big T. And then here we have big T, little t. So 50-50. But everybody's a tongue roller because every single offspring has a dominant allele. So just like Gregor Mendel predicted the idea of dominance and recessiveness, if that dominant little bit of DNA is in there and it tells you how to roll your tongue, right? It gives you the protein that goes underneath your tongue, then you're going to be able to roll your tongue. So since everybody has a dominant allele, they are all expressing the dominant phenotype, which is tongue rolling. Okay, so that's how to do that one. And then problem number 3, this is going to be the last one for this video. Oops, don't look. Go back and try this one. Go back and try this one. Our little hamsters. So give that a try. Hit pause. Give it a whirl. All right, so imagine you're back. So it says here that fluffy fur is dominant to smooth fur in hamsters. So fluffy is going to be the letter. We're going to use F's. Cross a smooth fur hamster. Okay, smooth fur. How the heck do I get smooth fur? What does that even mean? I didn't give you any words there. How do you do that? Well, if smooth is recessive, the only possibility for this hamster, the only genotypic possibility for this hamster would have to be little f, right? Because once it has a big F, it's going to have fluffy fur, right? So that was a little bit harder. They got a little bit harder as they went. So this smooth fur hamster has to be little f. And homozygous dominant, you should be good with that one. Homozygous means same, right? It's going to have the same alleles, but it has dominant, so it's going to be the capital letters. So this will be big F, big F. So really this problem is the same problem we've done so far. We've done this problem a couple of times already. When you cross two homozygotes, right, you always get hybrids. So here's the problem. Oh, let's clear that out. We cross big F with little f, you're always going to get 100% hybrids or heterozygotes. Right? So you can see there, 100% big F, that's the genotype. 100% fluffy, that's the phenotype. Okay. And just explain that. And then here's some more problems that you can check out on your own. A pea plant is dominant over, purple is dominant over white. What's the cross between two heterozygotes? Ooh, that's a good one. Two heterozygotes. Okay, so big P, little p, cross with big P, little p. Give it a try, hit pause. Okay, do that. You're going to get 3 purples and 1 white. Okay. All right, so that's the end of this video. These are all of these examples are examples of Mendelian genetics. The next video is going to cover non-Mendelian genetics, where I've said it, I probably said it like 12 times at this point. When the dominant allele is present, the dominant trait will be expressed. We're going to see that that's not always true. More times than not, it's not true for that dominant trait to be expressed when the dominant allele is present, because all sorts of interesting things happen. So we're going to see that in the next video. So this video is just about Mendelian crosses. All right. See you soon.